What's up, guys? Welcome to this lecture of genetic terms and concepts. So in this one, we're basically going to cover the terms that once you understand these terms and once you comprehend them, learning genetics will be much simpler and easier. So first we have chromosome. So chromosome is a long thread-like structure. It's made of DNA and proteins that carry genetic information. So here, as an example, it's a karyotype, and it includes all 23 chromosomes. We have chromosome number 1, 2, 3, and so on to 23. So obviously we have one from the maternal side, one from paternal, and these combined make up 46 chromosomes. So next we have gene. Gene is a DNA segment of a chromosome that encodes the instructions for making a specific protein. Here, for example, let's say this segment right here, it will make up this specific protein, and then there'll be a different segment that's going to make a different protein. Next we have genotype. Genotype is a genetic makeup of a person, and every single individual has a different genotype. That's what makes them different. Next, moving on, is phenotype. Now, phenotype is observable physical and biochemical characteristics of a person. So, the key point here is it's observable. So, for example, let's take your height. So, your genotype will probably code for, let's say, you to be 6 foot, but your phenotype will be your actual height. It's something that we can observe. And so phenotype depends on two things. The first one, obviously, is genotype, and the second one would be environmental factors. Let's think about it. Let's say your genotype encodes for you to be 6 foot, but you do not get proper nutrition. So what's that going to lead to is that's going to lead you to be, let's say 5'8" or 5'7" or whatnot. As you're growing up, you don't get your proper nutrition. So a good analogy for genotype and phenotype is a recipe. So let's say this right here is a blueberry muffin. So the recipe for this would be your genotype, but the actual muffin would be the phenotype. So next is locus. Locus is a specific location on the chromosome where gene is located. Think of it as an address for genes. So let's say, for example, this is your house and you have a friend right here. And then your friend says,
please just send me location.
So, and let's say this is your house number 123, and this is the street you're on is called Main Street. So for your friend to come over to your house, you have to give him your location, your address, which will be 123 Main Street, and then he's going to figure it out. He's probably going to put it in the GPS and make his way to your house. Just like that, genes also have an address. So let's take this gene right here, for example, CFTR gene. So the address for this gene, or the locus for this gene, is 7q31.2. So now what does that tell us? The 7 here tells us this is on chromosome number 7. The Q tells us it's on the Q arm, which is a longer arm, and the top one is P, and 31.2 points to this region right here. Next we have allele. Allele is different versions of the same gene at a specific locus. So let's say, for example, let's take an example with your blood type. Let's say this is the locus of your blood type. This This is the gene. And let's say this is maternal, paternal. So from mom's side, you get A. And from dad's side, you get B. Sorry, this is supposed to be a B. So yeah, from dad's side, you get a B. So these are the same genes coding for the antigen of your red blood cells, but they are different. They are different variations. And so these are different alleles of the same gene. Yeah, so these are different different alleles of the same gene. So now let's say this is in A and B. What is that called when they're different alleles of the same gene? That is known as heterozygous. Now let's say you had exact same copy. Let's say this was A and this one was also A. So they're same, right? What would that be? That would be, if this was heterozygous, this is homozygous. Okay, moving on to codominance. So codominance is both the alleles equally contribute to the phenotype of the heterozygote. Okay, so let's go back to the blood type. Let's say from this side, on this one it's A, and on this one it's B. So codominance tells us that these will equally contribute. So on red blood cells, you're gonna have antigens both for A and B. Next is variable expressivity. So this is patients with the same genotype, with the same genotypic makeup, have different phenotypes. So let's say both these patients, this one right here and this right here, both the patients have mutation in NF1 gene. What's that going to lead to? That's going to lead to neurofibromatosis. But variable expressivity tells us that this patient, for example, will show with cafe au lait spots, and he might have neurofibromas. But this patient might have leash nodules, and freckling. So the point is they have the same mutation, but they're showing different symptoms, different phenotypes. Next, incomplete penetrance. So not all individuals with the pathogenic gene will express the disease. For example, let's take the BRCA1 gene. All these patients, all these females have mutated BRCA1 gene. So this one develops cancer. This one develops cancer. But this one, although she has a mutated gene, she is negative for cancer. That's incomplete penetrance. Then we have Pleiotropy. This is when one gene contributes to multiple phenotypic traits. For example, let's say this person has mutated FBN1 gene. We know that's going to lead to Marfan syndrome. So the point is one gene, mutation in one gene, but you're going to see the effects of it on different systems. For example, you're going to see it on skeleton system. This is going to have an effect on the cardiovascular system and the ocular system and skin and lungs and whatnot. You know, all these mutations, all these phenotypic traits are coming from a single mutated gene, and that's the FBN1 gene. Next is anticipation. So anticipation is increased severity or earlier onset of disease in succeeding generations. So let's say this is the grandfather, this is the father, this is the son. And all of them have Huntington's. That's Huntington's right there. But so we know what happens in Huntington's is there's a repetition of the trinucleotide CAG, which encodes for glutamine, I think. So let's say this person has 40 trinucleotide repetition, and this person has 80, and the son here has, let's say, 100. So this person, he has 40, which is the more the repetition, like the more more severe the mutation is. So this person, the symptoms for the disease might show up when he's, let's say, 45 years old. And because this person has more repetitions of the CAG trinucleotide, meaning it's going to show up earlier. So let's say it shows up, the symptoms of Huntington shows up when he's 40. For this person, the repetition is even more severe. So what does that mean? It's going to show up earlier, let's say 35. That's anticipation. Loss of heterozygosity. So this is when one allele of a gene is lost. It's lost in a cell that originally had two different alleles. So let's say, for example, this is a chromosome, and this, let's say these are two different alleles, and this is allele 1, allele 2. Somewhere down the line, something happened, and this person lost one of the alleles. So what happened? That's loss of heterozygosity. So now this person has loss of heterozygosity. So what does that leave him with? Does that mean he's homozygous? Actually, since he only has one allele, there's another term called hemizygous. So this person is left with a hemizygous allele. See, now this concept is quite important when we discuss about tumor suppressors. So let's say RB, for example. So we know in order for tumor to form or cancer to form, both the RB genes have to be either mutated or deleted or silenced. So let's say let's take an example. This person has one RB gene right here this is wild type, or also known as normal. Let's say this is normal, but this person, this allele is mutated. So will the patient show up with a cancer? No, because we need both the genes to be either mutated or deleted or whatever. But now let's say loss of heterozygosity occurs and he loses the normal allele. That obviously will lead to cancer. So next is epistasis. Epistasis is a gene that is influenced by another gene. So, for example, let's take two different genes right here, TYR and OCA2 gene. So TYR gene helps with the production of melanin, and melanin will determine the complexity of your skin, if it's going to be fair or you're going to have a darker skin or fair skin, and that is decided by OCA2 gene, which helps regulate the amount of melanin being produced. So for example, this person, the TYR gene works perfectly fine, and the OCA2 gene regulates that this person, because he's fair complexion, lighter skin, is going to have decreased melanin production. for this person, let's say his TYR gene works completely fine, but melanin is increased, decided by OCA2 gene, right? I'm sorry, I mean like the amount of melanin produced is increased. But this person, he suffers from something known as, so in this person, the TYR gene is mutated. So regardless of the amount or the regulation of melanin that was to be done by the OCA2 gene is pointless. So this gene right here, because of this gene right here is influenced, the function of this gene is influenced by the TYR gene. Or in other words, the TYR gene is epistatic towards OCA2 gene. Next we have aneuploidy. Is an abnormal number of chromosomes. So we know normal amount of chromosomes is 2. This is normal. But if we have 3 chromosomes, we know that is abnormal. And if we have 3, that is known as trisomy. If 3 is trisomy, so 1 will be monosomy. So in this case, for example, let's say this is chromosome number 21. We know that's going to lead to Down syndrome. And let's say this is a single X chromosome and the other sex chromosome is missing. So that is something known as Turner syndrome. But yeah, so trisomy and monosomy are both examples of aneuploidy. Next, we have dominant negative mutation. This is an altered gene produces a non-functional protein that also prevents the normal gene from functioning. For example, let's take P53 here. So let's say this is a P53 protein. So we know P53 usually works as a tetramer right here. So when a tetramer forms, P53 can then go and attach to its receptor, which will, if you've covered the previous lecture on cell cycle, you know it's going to halt the cell cycle. That's the job of P53. But let's say there is single this protein right here is altered, but the other three, the other three P53s are normal. But there's only one that's been altered. So now, do you think this can connect or attach to its receptor? Absolutely not. So this one gene, this one protein, it altered, it changed, it prevented the whole tetramer from functioning. Next, we have mosaicism. This is presence of genetically distinct cell lines in the same individual. So you have more than 2 or more than 1 cell line in your body. So let's say when the sperm fertilizes an egg, we know it's going to start to replicate, and let's say there is a mutation that occurs here. As you can see, this represents a mutated cell. So obviously it's going to go ahead and replicate more and more and more, and eventually it's going to have few mutated cells. So this represents the mutated cell line. So when a person will be born, you can see as here, it will show on his skin. This right here will be the mutated cell line, and this will be normal, as you can see in the rest of his body. Here is an example, is a good example of that. This is a disease known as McCune-Albright syndrome. As you can see, this right here is the mutated cell line. And this one right here is normal. You can see right here, this spot right here, these spots are all mutated. Next, we have Locus heterogeneity. this says mutations at different loci result in the same disease. Different loci, same disease. Okay, let's say, for example, this is chromosome number 1, chromosome number 3, and this is X chromosome. Let's say there's a mutation here on a gene called RHO. We know that mutation will lead to a disease called retinitis pigmentosa. And let's say this is chromosome number 3. And on chromosome number 3, there is another gene right here. Let's call it RP9 gene. And any mutation to this will also lead to retinitis pigmentosa. And then on X chromosome, there's another one. Let's call it RPGR. And let's say that mutation is here. That mutation is right here. That will also lead to retinitis pigmentosa. So mutations at different loci, same disease. Next, we have allelic heterogeneity. This one is different mutations in the same locus leads to same disease. For example, let's say this is CFTR gene, right? This whole thing. For example, let's say there's a mutation here. So what is that going to lead to? Mutation in CFTR gene leads to cystic fibrosis. And now let's say there's a different mutation in the same locus. There is a different mutation. Guess what? That leads to cystic fibrosis. So this is all single locus, a single gene. So different mutations in this, whatever mutations occur, it's going to lead to cystic fibrosis, the same disease. Next, we have heteroplasmy. Heteroplasmy, this is presence of both normal and mutated mitochondrial DNA. So that tells us in a single cell, they're both mutated. These are all mutated mitochondria, and we have presence of normal. So if heteroplasmy is a presence of both normal and mutated, so what would be the opposite? The opposite would be homoplasmy. So that homoplasmy would mean either all your mitochondria are mutated or all are non-mutated. They're normal. Next, we have imprinting. So imprinting is one gene copy is silenced by methylation and the other copy is expressed. So imprinting is a normal phenomenon that occurs. For example, let's say this is chromosome number 11. On chromosome 11, there is IGF2 gene that, and let's say this is the maternal side, this is the paternal side. Usually, the maternal side is imprinted and the paternal side will be expressed. So that's normal. But if the expressed gene, the one that is supposed to be expressed, is mutated, then that could lead to pathologies. It's also known as disorder of imprinting. So let's take, for example, right here, this is chromosome number 15. So in this one, the paternal gene right here is imprinted, right? Let's say this is the gene, paternally it's imprinted. And the one on your maternal gene is supposed to be expressed. But let's say the one that is supposed to be expressed gets deleted. That will lead to a pathology. And that's called Angelman syndrome. So let's take another example. This is chromosome number 15. And let's say there are a few genes that are, these right here are supposed to be imprinted. Right, there are a few genes that are supposed to be imprinted here maternally. And on the paternal side, it's supposed to be expressed. Say this side is supposed to be expressed. But let's say these genes that are supposed to be expressed get deleted. Now what will that lead to? That will lead to something known as Prader-Willi syndrome. Next, we have uniparental disomy. This is when offspring receives 2 copies of a chromosome from a single parent and no copies from the other parent. So we know usually let's say this is Bob right here. So we know Bob's gonna get 2 chromosomes, one coming from maternal and one coming from paternal. But let's say instead of getting one from paternal, he gets both from maternal. Then that this person has uniparental disomy. Thank you very much for watching this video, and if you found it helpful, make sure to like and subscribe and share with someone who might be struggling in this topic. Go make their day.